This is a representation in pi-mole of the protein phenylalanine hydroxylase. Looking at the primary structure, there is a sequence of 335 amino acids, the building blocks of protein, and they fold up based on two main principles: hydrogen bonding between peptide bonds, making secondary structure, and the type and orientation of bonding between R groups, making tertiary structure. You can't see R groups or bonds being formed here, but you can see the structures that arise from them. Amino acids 1 to 117, aka the regulatory domain, are also not featured here, as the regulatory structure is not so well understood structurally. But what you do see here is the catalytic domain and tetramerization domain of phenylalanine hydroxylase. The catalytic domain here is involved in acting on the phenylalanine, converting it to tyrosine, and the tetramerization domain here in dark red is involved in joining four polypeptide chains together. And if we look at the protein from a surface view, we can see here in dark red, that is where that tetramerization is occurring. Now we can see here that this addition of the protein to PAH, it's comprised of two polypeptide chains, one blue and one green. These are very similar amino acid sequences. Each of these chains contains 13 alpha helices, shown here in light blue and light green. Their function is to do with creating a hydrophobic active site for phenylalanine. The model also contains 3 beta barrels, so 6 beta sheets on each chain. 2 are indicated in orange here, one there, and then in dark grey, there's another one here. It's interesting to note here that when only one polypeptide chain is present, such as in this representation of PAH, this one here, the secondary structure of this protein contains 8 beta sheets and 4 beta barrels. So we can infer that by calling on the tetramerization domain to create a tetramer, that is a protein comprised of 4 polypeptide chains, a beta barrel is lost. Again, we can see 3 beta turns present in this structure, here, here, and here. And if we look at these closely, we can see a significant change in direction between two secondary structures of the polypeptide chain. And embedded in the center of the protein, an iron atom known as an iron ligand, and this has a crucial involvement in tyrosine synthesis.